Hello everyone. In this video, we'll begin our analysis of proton NMR spectra, or H1 NMR nuclear magnetic resonance. So we'll begin by identifying the number of signals that exist within a compound. So if we're given a structure, we can figure out essentially how many different signals are going to appear on the NMR spectrum. And then we're going to further analyze those individual signals. So when we look at the sort of analysis of those signals, we can figure out information by where those signals are located on the x-axis of our spectrum. The integration, so essentially how many hydrogens each of those signals represent, and also the shape of those signals. So the shape of the signals will tell us essentially the surrounding environment. So at the end of this video, hopefully we'll have at least a beginning idea of how to take a look at a proton NMR spectra, and we'll also practice taking a look at identifying the number of signals, looking at a compound. So if we take a look at this representative spectra here at the bottom, notice that we look at the spectrum and we can identify that there are three overall signals. So what I mean by a signal here is basically wherever there is a peak, a unique peak that exists along the x-axis. So notice that I have this signal here, and a signal at this position here. It's important to identify the location of those signals. So we're looking at the x-axis and we're calling that essentially the chemical shift. And the units for that is in ppm. We'll explore what that means in just a second. But ultimately, we want to be able to identify where the peaks are located. And similar to our spectroscopy, there are certain regions of our NMR spectra that correspond to specific types of hydrogens. So it's important for us to be able to locate and identify what sort of unique peaks can occur within those particular regions. And we call that the chemical shift. Next, we'll take a look at the integration. So if we take a look at each individual signal, so again, A, B, and C, by identifying and doing some math and solving the integration, so solving the area under each of these individual peaks, it can tell me essentially how many hydrogens are represented by that given signal. So for signal A in our spectrum, we can see that there are 5 protons or 5 hydrogens that exist within that spectrum or within that signal. For signal B, there are going to be 2 hydrogens, and for signal C, there will be 3 hydrogens. Now, we're not necessarily going to have to, do the math to solve the area under the curve, but we'll be given some sort of information that can help us to determine the number of hydrogens for those individual signals. And then lastly, when we're taking a look at our signals, we want to take a look at the shape of the signals, because that provides some idea as to what the surrounding environment of those protons will look like. So essentially what this does is it tells me what are the adjacent hydrogens to our chemical signal. So we call this the splitting. So we can identify that we would say that signal A has a fairly complicated splitting. So there's a lot of different peaks. It's kind of different heights of those peaks. If we look at signal B, this is a little bit more regular. So notice that there's 1, 2, 3, and 4 peaks for this particular signal. And for signal C, there's 1, 2, and 3 peaks. And again, that can tell us information about the adjacent hydrogens to our signal. So now let's take a look at identifying the number of signals. And that's usually going to be where we start when we're analyzing an NMR spectra. So number of signals is really dependent upon the structure of the molecule and ultimately the chemical environment that these hydrogens exist in. So if I take a look at this simple alcohol, so methanol, notice that I'm going to essentially have 2 different hydrogens that exist. So we have the hydrogens that are attached to my carbon here, and I have a hydrogen that is attached to the oxygen. And so the hydrogens that are attached to the carbon will feel the overall magnetic moment. So again, we're putting the sample within a fairly large magnet and exposing it to that external field. So when we say that this is shielded, there's sort of an electron environment around this that is essentially requiring us to use a little bit more energy to promote that alpha spin state to the beta spin state. So if it's shielded, it's going to require, again, a little bit more energy to promote that spin state. If it's de-shielded, there's sort of less electron density around that proton. It's going to require a little bit less energy to promote from that alpha state to that beta state. And so this basically, by having slightly different chemical environments, that puts us in slightly different portions of our x-axis on our proton NMR spectrum. So if we take a look at the less shielded, so in this case, our hydrogen here, we would say that that is further downfield. So it'll require a little bit less energy to promote that alpha, the promotion to that alpha to that beta state. Whereas the ones that are more shielded are again going to require a little bit more energy, and those are sort of further upfield or further to the right on our NMR spectrum. So the important part here too is to also notice that we have two signals for this particular compound. The reason for this is we say that the three hydrogens that exist on our carbon are chemically equivalent. So if we look at sort of the bonding pattern here, all of the hydrogens have the same sort of relative position to the oxygen. So they're all bound to this carbon, which are bound to this oxygen. So all three of these hydrogens will exist in the same signal on your proton NMR spectrum. So all three of the hydrogens exist within this signal here. That is partly why we have different heights for these overall signals. We'll explore that a little bit more when we talk about integration. So now what we want to do is when we're taking a look at the structure, we want to be able to identify chemically equivalent hydrogens. So what we do is first analyze that spectrum and figure out how many different sets of hydrogens will exist within that compound, and that can tell us the number of signals within our proton NMR spectrum. So if we look at this alkane here, notice that we have a 3-carbon chain, and there's actually a plane of symmetry. So the plane of symmetry is going to exist down that central carbon. So if I were to draw the plane of symmetry here, it's a little bit off, I apologize, but down this central carbon here, the hydrogens attached to this carbon are identical to the hydrogens attached to this carbon here. That is what we would say that they are chemically equivalent. And so these hydrogens in the middle carbon are going to be different. So if we put that all together, we are going to have two different sets of hydrogens and therefore two signals in our proton NMR spectrum. If we look to our ether, again, we have a plane of symmetry directly down our oxygen here. So if we were to go down the oxygen here, there's our plane of symmetry. So the hydrogens on the carbons that are attached to the oxygens are chemically equivalent. So these hydrogens here and these hydrogens are chemically equivalent, as are these hydrogens and these hydrogens. So again, if we put that together, we have two sets of hydrogens and therefore two signals on our proton NMR spectrum. If we look at our ester, or excuse me, our ester here, notice that there's no plane of symmetry. So all of our areas of hydrogens are going to be different. So we have 1, 2, and 3. So we're going to have 3 sets of hydrogens and therefore 3 signals on our NMR spectrum. Similarly, our alcohol has no plane of symmetry. We have hydrogens on this carbon here, and this carbon here, as well as the hydrogen that is attached to our oxygen. And therefore, we will have 4 signals in our proton NMR spectrum. So now what I would do is pause the video and try to solve these quickly, and then come back and listen to the explanation after you have completed. Okay, so let's walk through these particular examples. So this ketone here, again, has no plane of symmetry. So we'll identify all of the carbons that contain hydrogens. There's 1, 2, 3, and 4. All 4 of those signals will be different. So we have 4 sets of hydrogens and therefore 4 signals in our proton NMR spectrum. If we look at this ketone here, this one does have a plane of symmetry. Again, we can draw that plane of symmetry right down that carbonyl carbon. So again, thinking about the relative positions, the carbon here has hydrogens and the carbon here are in the same chemical environment. Again, they have the same sort of bonding to that central carbonyl. And these 2 carbons here with hydrogen and the hydrogens here are chemically equivalent. So there will be 2 overall signals. The important part here is that we can utilize proton NMR spectroscopy to differentiate between these 2 constitutional isomers, these 2 ketones. If we take a look at this ring here, so we have hydrogens on all of these carbons, but all of them are chemically equivalent. So we have lots of symmetry, so technically 5 planes of symmetry for this. So all of the hydrogens will be in the same chemical environment. So there would only be one signal in our proton NMR spectrum. If we take a look at this alcohol, notice that we have hydrogens on all of the carbons at the end here, and all of those hydrogens are the same distance away from this carbon and away from this oxygen. So we would say that all of these hydrogens on these carbons are chemically equivalent. So that is one signal. And then we have our second signal from our hydrogen on our oxygen. So there would be two sets of hydrogens and two signals in our proton.